My today's discussion topic is the nutritional manipulation to combat the nematodiasis. So grazing-based livestock husbandry plays an important role in the rural economy of India and its neighboring countries. And in other part, the gastrointestinal parasitism is a major problem in the small ruminant production worldwide due to its impact on animal growth and productivity and the associated cost of control measures. This problem of this gastrointestinal parasitism is of particular importance in developing world because in the developing world, the nutritional resources available for this livestock are often inadequate. Due to this, acquired immunity is often compromised, resulting in lowered productivity and increased mortality. The nutritional status of the host has a significant effect on the ability to withstand the parasite infection, and it has also been frequently observed that the mortality resulting from these nematodes is more severe in the malnourished animals than in well-nourished animals. In the different coproculture studies of these sheep and goat fishes, here we can see that Haemonchosis contortus, that is the main gastrointestinal nematode having 70% of the positive cases, and then Trichostangulis columbriformis, that is 20%, and Strongyloides papiliosus, that is 10%. So this Haemonchosis contortus is the largest GI nematode, measuring 20 to 30 mm, and the female parasite is a prolific egg layer, producing thousands of eggs each day. So Haemonchosis contortus, this parasite is harbors in the abomasum of affected animal and feed on its blood of the host, producing mild to severe symptoms and even death in acute form. This parasite that cause heavy production losses and is of that's why the economic importance. Especially this Haemonchosis has developed diverse characters over the years, leading to limited success in the production of the vaccines. Coming to the life cycle, this parasite is having 6 stages of life: egg, then the 4 larval stages, and the adult. The female parasite may lay a numerous egg, that is an average of 1295 per day, which are passed out through feces to the pastures. So that means they are laying a huge number of eggs per day per parasite. This egg may die or develop to free living larval first stage, that is L1, L2, and then infective stage L3 within a very short span of time, that is 1 to 7 days. This L3, the hatchability of the eggs and the development of infective larvae depend on the availability of suitable environmental condition, because this will take place in the pasture or in the harvest. So this suitable conditions, it rise from the 15 to 37 degrees Celsius of temperature, and then humidity of 85 to 100%, which is very much prevalent in our condition or in our Indian environment. This stage L3 is ingested by the host, where it undergoes encystment in the rumen and takes 2 to 3 weeks to develop to the parasitic stage, that is the L4 stage. Here 2 times molting occurs, and just before the final molt to L5, it erupts, which develop a lancet to penetrate the mucosal vessels for sucking the blood. Their main predilection site is the abomasum, where the adult worms move freely in the fluid. The parasite may also undergo arrested inactive phase of development in the host during winter, called as the hypobiosis. So they can stay in the winter for a prolonged period or for the entire winter period, and then whenever the summer or some rainy season starts, or the environmental condition occurs, so from there they can proliferate and go for higher incidence of the parasitism. So here we can see the different photographs, where first is the egg that is we are tested in the fecal sample. Then next is the L3 larva with the tapering end. It is the characteristics of this Haemonchosis contortus. Then the adult female, it is the vulvar flap I am showing in the third photo, that is C. And in D, we can see the adult male having the copulatory bursa and the spicules. For this eggs is transmissible in a most suitable condition is that the larvae in the majority of helminth parasite are provided by warm, wet weather. Here, the larvae can remain alive for 6 to 8 weeks. The hot, humid tropical climate of our country is suitable for development and survival of pre-parasitic stages of these nematodes throughout the year. They can sustain, if they are not getting a suitable host in the pasture, they can let live, or they can go for continuing in that L3 stage for a year also. So these young animals and the animals raised on poor diet are more susceptible to parasitic infection and less able to mount a curative or protective immune response. The animals that are stressed, immune compromised, or suffering from a concurrent disease are more likely to show the signs of the parasitic diseases. Diseases and parasites have a heavy impact on the kids because of the poor immunity status of these young animals, leading to an increased susceptibility. Coming to the types of different form, that is, first is the hyperacute cases, that here we'll see the sudden death. And in acute form, it involves the anemia, lethargy, weakness, increased respiratory and heart rate, dark massive feces, loss of wool, pale to white conjunctiva, ascites, and submandibular and cervical edema. In chronic cases, we'll see the anorexia, loss of weight, agalexia, failure of the conjunctiva, and mucosa. Coming to the pathogenesis and symptom, degree of the establishment of this parasite and the consequent sickness depend upon the number of infective larvae ingested and the age of the host, immunity level, and nutritional status. The major pathogenic damage occurs due to the sucking of the blood by the free-living parasites and the eruption of ulcerative lesion in the abomasum mucosa, leading to digestive syndrome and anemic disorders. Infected sheep that may lose up to 30 to 50 microliter of blood every day due to one parasite. Single parasite can suck blood of 30 to 50 microliter. And there are more than thousands laid by one parasite, one adult female parasite only. So even death in the prepartum period also occurs. So loss of blood, which is either ingested or let oozed out from the mucosal lesion to feces, leads to anemia, which appears 10 to 12 days after getting infected. And at the 4th day itself, the packed cell volume is also detectable. There is less PCV is detectable at 4th day. This PCV value may drop by 3 to 6 weeks up to a level which is due to accelerated activity of parasites and bleeding from the hemorrhagic gastritic lesions. Then this concurrent reduction of the PCV along with the concentration of the hemoglobin and plasma protein is also observed. This extensive damage to the abomasal mucosa affects the rate of passage of the ingesta, then produces pain and inflammatory cytokines and changes in the gastric secretion as well as level of the gastrointestinal hormones in the plasma, which lead to prolonged loss of appetite. Thus, the animal go for lowered feed intake. Due to this, raised pH of the abomasum occur, and rumen microbes do not get inactivated and lysed, resulting in non-availability of the amino acid or the non-availability of the microbial protein. Physical and chemical damage that has been caused by the parasite induces the inflammatory response in the gastric tissues, leading to collection of numerous neutrophil lymphocytes and eosinophils, which further aggravate the situation. Coming to the control of these parasites, the indiscriminate use of the anthelmintic in the field condition has produced drug resistance against almost all conventional products. So due to this constantly increasing drug resistance in this parasite and residual effect of the anthelmintic, the control strategies which employ minimal use of synthetic anthelmintics have gained the importance. Many experts have recommended an integrated control mechanism encompassing various approaches instead of relying on a single option to achieve the enough control. The managemental conditions like selection of resistant lines of the sheep, adoption of grazing techniques, and vaccination program has also gained significant attraction. Different types of medicinal plants and non-conventional chemicals have also been reported to be used in certain controlled experiments. However, the research on that medicinal plant and non-conventional chemicals, their adverse effect on growth and fertility is yet to be studied. Then next, research on the molecular tools for identification and the introduction of resistant genes into the flock is also underway, but there is a still long journey to find its field applications. In given circumstances, a targeted selected treatment approach, along with selective breeding, culling of more susceptible animals, and maintaining a good body condition score through the provision of balanced diet remains a workable strategy to control the haemonchosis. To maintain this body condition score, a good one, we have to go for some nutritional manipulation. So the manipulation of host nutrition is the alternative method to anthelmintic to improve the host resistance and the resilience to parasitic infection. It is the most promising option to reduce this dependency on chemotherapy or conventional chemotherapy and to favor the sustainable control of gastrointestinal nematode infections. This manipulation or the rationale for this manipulation of nutrition to control the GI nematodes is based on knowledge of pathophysiological processes associated with this presence of worms in the gut. This worm generally induces decreased appetite, decreased digestibility of food, and the diversion of nutrient from production sites towards the repair of the damaged tissue and the homeostasis, causing the assimilation of nematode parasitism to a nutritional disease. Adult parasites that has been found attached to the abomasum mucosa or freely moving in the mucosal fluid. Female parasite is hematophagous and causing a loss of both blood and the protein. The developmental stages occurs within the gastric gland of the fundic region of the abomasum, leading to formation of visible nodules on the abomasal mucosa and causing extensive deformation of the epithelium and subepithelium, leading to chronic inflammatory process which compromises the nutrient absorption and may lead to severe diarrhea and weight loss. This resulting hyperplastic gastritis disrupts the secretion of gastric enzymes and alters the gastric pH, which turn leads to the clinical symptoms of the diarrhea, dehydration, and the weight loss. So these two photographs I'm giving here is a case of in a farm where I have seen the typical lesions of the Haemonchosis parasites in a goat. This is to the abomasum of goat, where in the first photo we can in the left side we can see the abomasum is having some hemorrhagic conditions or the red spots, and in the second photo in the right side we have seen that nodule formation in the which disrupts the epithelium also. So this is the typical reason, and in this condition I have seen the entire flock is having low hemoglobin content. By that PAMASA test we have done that, and we have seen the pale mucous membrane of the eye and all those other symptoms. And in PM condition, when the animal, 1 or 2 animals died, and we have done the PM, and in that PM we have seen this typical heavy conditions of the haemonchosis. So there is reduction in the local immunity also, such as the lower antibody levels and fewer mast cell, globule leukocytes, and goblet cells in the intestinal tissue occurred in both pregnant and lactating ewes. These observations suggest that the changes in local immunity conditions, particularly the IgA level, may enhance the parasite egg shedding, which indicated that parasite-specific IgA negatively impacts the parasite fecundity. So here the improvement of the host diet has been associated with two distinct potential benefits. From here you can see that firstly, the host diet will provide the nutrient needed by the increasing demand to raise an immune response against the worm. And secondly, it will contribute to maintain the tissue and the blood homeostasis and the host production despite the presence of worms. So keeping all these points, does an improvement in host nutrition, we can tell that it will enhance the host resistance, that is its aptitude to regulate the worm population, as well as the host resilience, that is ability to withstand the negative effect of the nematode infection. If you go for the pathophysiological study, we can see that the protein metabolism is much more disturbed by these GI nematodes than the energy metabolism or the energy component of the diet. The animal need protein to build and repair various body structures such as muscles and for the immune response to various infections, including the worm infection. Protein supply, it comes from two sources in case of grazing animal. One, the microbial protein, that is the rumen microorganisms will convert the plant protein consumed by the animal to microbial protein. And the other one is the some protein which are not been degraded in the rumen. It will be go to the small intestine, that is the bypass or protected nutrients, we can say, and that will be directly taken up by the host animal. Then this supplementation of the feeding of young both with protein and high quality pastures can increase their resistance to worm infection and production in worm infected animals. If the nutritional demand of infection, that is the blood sucking parasite, whatever they are doing, the direct loss of nutrients by sucking the blood and along with the nutrients are met, then additional nutrient will be used for the muscles. Thus, the increasing the protein content of the diet resulted in the reduced fecal egg count and the worm burdens. The parasitism of the abomasum as well as the small intestine causes extensive protein losses, and they redirect the protein synthesis away from the skeletal muscle into the repair of the gut tissues, leading to reduced nitrogen retention. So this diversion of nutrients from production process towards the responses is essential for maintenance of homeostasis, such as plasmatic protein synthesis, mucus production, repair of the gut, immunocompetence. These are privileged over the functions that over the normal functions, that is the skeletal growth, soft tissue deposition, milk production, etc. So this nutrition, in particularly the protein nutrition, can have a major effect on both the resistance and the resilience of the animals to the parasites. The gastrointestinal parasites is having the main effects on erythrokinetics, plasma protein metabolism, and protein turnover in both skeletal muscle and liver, leading to development of anemia and the hyperalbuminemia. So they are associated with high fractional rate of removal of RBC and the albumin, which are attributed to the elevated enteric loss of protein at the site of infestations. So this leakage of protein into the gut was to increase the rate at which the hemoglobin and the albumin were synthesized and compensated for the losses in blood plasma. The increased rate of protein synthesis, which is required by the animal to go for resistance and the resilience, will require more amino acid. The significant loss of amino acid through the process of deamination and fermentation also will reduce the availability of amino acid to the animal. This all imbalance in the protein metabolism will go for decreased feed intake. And simultaneously, the energy metabolism is largely dependent on the utilization of volatile fatty acid. In case of these ruminant animals, they are also being hampered. In these conditions, in these parasitic conditions, the homeostatic regulation, that the blood glucose is also having moderate variability, and therefore has an intermediate diagnostic value in the assessment of nutritional status of the goats. The lower feed intake that also results in decreased blood level of propionate and glucose precursors, which are very much important for the energy metabolism, derived from the diet, decreased resulting in a reduction of rate of glucose synthesis also. The growth rate of animals influences glucose requirement, whereas in mature animals, only maintenance energy is required. So thus the growing animals or the kids have been mostly used with this GI nematodes. The physiological status of an animal, which is also important to combat this nematode infection, is also has an effect on glucose requirement depending on energy requirement for production. Dietary deficiency of the minerals like cobalt, copper, phosphorus also led to reduction in the animal resistance. This parasitic infection involves severe blood loss and inducing disturbance in iron metabolism could produce a secondary iron deficiency anemia. So young sheep with subclinical infection of T. columbriformis utilize calcium and phosphorus inefficiently has been shown by different researchers in their different works. This abomasum and intestinal infections adversely affect the deposition of calcium, phosphorus, magnesium in the growing animals, which cause reduction in bone size, density of the bone matrix, and degree of mineralizations of the bone. The parasitic infection did not affect the absorption of calcium and phosphorus. However, it reduces the bone mineralization and growth and leading to matrix osteoporosis. Thus, the balanced mineral supply is needed for the protection or for improving the immunity against the nematode infection. So main effect of the GI nematode infection is mineral and trace element absorption. That is, reduction of the mineral and trace element absorption takes place in case of GI nematodiasis. And it is maybe due to the gastroenteritis or which may affect the bone growth also. Thus, it will ultimately affect the growth of the animal. The absorption and retention of phosphorus is severely reduced. There exists an endogenous loss of calcium and reduction in absorption of this mineral in different studies have also been shown by different researchers. Thus, well-balanced and nutritionally supplemented diet we have to give in sufficient quantity to the sheep and goat during their late pregnancy or in case of growing animals. So this quality nutrition, it adds the expression of resistance and resilience of the host. Diet supplemented with high protein or the amino acid like methionine and lysine and rumen protected protein have been shown to boost the immunity, decrease parasite proliferation, maintain production, and reduce fecal egg count under infective conditions. So after going through all the points or after going through all the conditions, thus we can say that high protein level on adult productive in tropical conditions allows the reduction of the effect of gastrointestinal nematodes during the production period. This effect of dietary protein and reduction of the fecal egg count has been attributed to a replacement of nutrients that are diverted to develop the protective element of the immunity. So protein losses due to the GI infection can be corrected with the intake of dietary protein, and for this reason, protein requirements are higher than the currently recommended for non-parasitized animals. High level of rumen-soluble protein in pasture also will result in increased ammonia concentration in the rumen. This ammonia concentration in the rumen or high ammonia concentration in the rumen may have the adverse impact on the rumen microbial population, and there is potential for synthetic subclinical and clinical ammonia intoxication where the crude protein in diet exceeds 14% in case of sheep. So researchers showed that protein supplementation reduced the level of parasitism in lambs, and also that high protein diets increase the production of ammonia in rumen digesta. This accumulated ammonia in the rumen might directly influence the rumen microenvironment by exhibiting the toxic effect on Haemonchosis contortus. So researchers are giving a conclusion that this high ammonia level will facilitate the removal of the Haemonchosis contortus. Adult female will be released in the feces due to the toxic environment of the ammonia. High rumen ammonia will also increase the pH of the rumen, which may also affect the excitement of the infective larvae. So there are some non-conventional compounds also, such as copper oxide and condensed tannin. These are also have been used extensively to combat this nematodiasis problem. The copper oxide wire that is initially used as a mineral supplement are now well known to be effective against Haemonchosis contortus in weaning lambs without any toxic effect. That condensed tannin is having in the various forest plants also have the capacity to control the GI parasites in case of SIP. Then next is the neem tree, which is effective against the Haemonchosis contortus under laboratory conditions due to its polyphenolic flavonoid content. Now, next is the saponin extracted from the forest plant. Whatever the saponin is present, they have also significant effect on the Haemonchosis contortus eggs or the population of the Haemonchosis contortus. The SIP experimentally infected with Haemonchosis contortus and supplemented with yeast Saccharomyces cerevisiae showed a significant reduction of larvae and a higher number of circulating antibodies. So now, in a nutshell, we can tell that there are two phases, that is endogenous phase and exogenous phase of the parasite, where we can see the parasites in the animal the adult parasite will lay eggs. That egg will come to the pasture, and from the pasture, due to the grazing by the animal, they will pick up that infective larvae, and that infective larvae will go to the abomasum, and there they will molt to L5 stage, where again the parasite will be able to lay eggs, and there is multiplication of the parasites. So here we can see the different techniques like rotational grazing, then use of the nematophagous fungi, then deworming with the selective animal, but highly prone animals will be dewormed. Then different unconventional plant extracts can be used to combat this GI nematodiasis. So actually this entire concept of work is from my MBSc research work, where I have taken local non-descript goat kits in Chhattisgarh with natural subclinical nematodosis. Means that is, they are having already the nematodosis from the grazing conditions, and they are reared under semi-intensive condition, that is grazing for 4 hours in the morning, and then they are kept in a confinement and feeding only one time, that is at the evening 3 o'clock. I have been given with the concentrate feed to the animals. The grouping is such that my main intention was to see this program that normally our Indian farmers, they are doing the goat or sheep farming in grazing conditions. So that's why I want to take the grazing part also, because the parasite is having mostly been transmitted while grazing in the infected pasture. That's why I taken the concept of keeping the grazing things also. So the one group I have taken solely, they are reared in grazing conditions in a good pasture. And then I have given different level of protein in the concentrate feed, whereby the level of protein more than the recommended level that has been given by our given in our feeding standards, the recommended level, the higher the protein I am giving, I am getting the better result of to combat these nematodosis conditions. So my result have shown that there is increased feed intake, body weight gain, feed conversion efficiency, blood hemoglobin, TC, TLC, and all the hemoglobin related parameters. Then serum protein, serum glucose, microbial protein, these all are in increased amount in case of high protein supplemented group. And there is lower egg per gram of feces, lymphocytes, and the eosinophils, then the other SGPT, SGOT enzymes in high protein supplemented group. The protein is given more than the normal requirement of the animal. So upon getting all the things or my personal experience from my research work also, so we can go for the conclusion that haemonchosis, it's a major problem in this small ruminant industry across the world. This organism has adapted to survive in the diverse climatic conditions. The resistance of the anthelmintic, whatever are available in the market, is already been reported. Alternative compounds have a limited efficacy, and their extensive study has not been done till now. So their effects on production parameters are least known. Biological control measures have their constraint because biological measures are always there is some disadvantages or to maintain the condition. So due to the diverse antigenic strains of the parasite, it has diverse antigenic strains for which the vaccine production is also a very much great challenge to the researchers. All these conditions, we can tell that targeted selective treatment, along with culling of more susceptible animals and maintenance of good body conditions score through the provision of nutritionally balanced diet is considered the best control strategy at the herd level. And at the meantime, the scientists engaged in its related fields should carry out extensive research for the development of a potent vaccine against all the Haemonchosis contortus strains and find their therapeutic agent, which could remain effective for decades without having a residual effect. Should we worry for these nematodes, or should we worry for this anthelmintics resistance that has been occurred in the field condition? So my answer is no, we should not worry about the anthelmintic resistance or this nematodiasis, but we have to be concerned about the use of these anthelmintics or about the conditions of the GI nematode that has been happening due to the extensive uncontrollable multiplication of the nematodes, and thereby we have to be concerned about their nutritional status, and we have to be concerned about the supplementation with the highly balanced nutritional diet. Thank you.